Ever notice how the MCAT loves turning something as straightforward as cell division into a maze of tricky questions? If G1, S, G2, and mitosis start to blur together, or checkpoints feel like random buzzwords, this is where we fix that. In the next few minutes, you'll build a clear, step-by-step mental map of the cell cycle, understand what actually happens in each phase, and learn how to spot the patterns the MCAT tests over and over again. The cell cycle consists of interphase, which includes G1, S, and G2 phases, followed by the mitotic phase. And understanding what happens in each stage is a classic MCAT target. In G1, the cell grows, synthesizes proteins, and carries out normal metabolic functions. This is often the longest phase, and where cells decide whether to divide or enter G0, a quiescent state. A helpful mnemonic is G1 equals growth 1. Next is S phase, where DNA replication occurs, doubling the genetic material so each chromosome now consists of 2 sister chromatids joined at the centromere. Think S equals synthesized DNA. Then comes G2, another growth phase, where the cell prepares for mitosis by producing microtubules and checking that DNA replication was successful. G2 equals growth 2 and get ready. Interphase as a whole is about preparation, not division. The mitotic phase includes mitosis and cytokinesis, where the nucleus divides and then the cytoplasm splits, producing 2 daughter cells. On the MCAT, you may be asked to identify which phase a cell is in based on DNA content. Cells in G1 have 2N DNA. Cells in S are between 2N and 4N, and cells in G2 and mitosis have 4N DNA. Another common angle is distinguishing interphase, where chromatin is uncondensed and not visible as discrete chromosomes, from mitosis, where chromosomes are condensed and visible under a microscope. Connecting this to other topics, DNA replication in S phase ties directly into molecular biology concepts like DNA polymerase, leading and lagging strands, and proofreading. Overall, knowing the sequence G1, S, G2, M, and what each phase accomplishes is foundational for interpreting experimental data and passage-based questions. Cell cycle checkpoints, particularly at the G1 to S transition and the G2 to M transition, act as quality control systems that ensure the cell only proceeds when conditions are appropriate, a concept heavily tested on the MCAT. The G1, S checkpoint is often called the restriction point and asks, is the cell large enough? Are nutrients sufficient? And is the DNA undamaged? If the answer is no, the cycle pauses or the cell enters G0. The G2M checkpoint verifies that DNA replication in S phase has been completed accurately and without damage before mitosis begins. A useful way to remember this is G1 checks the environment, G2 checks the DNA. These checkpoints are controlled by cyclins and cyclin-dependent kinases, or CDKs, which form complexes that drive the cell cycle forward when activated. Tumor suppressor proteins, like p53, play a crucial role by halting the cycle if DNA damage is detected, allowing for repair or triggering apoptosis if the damage is irreparable. This ties directly into cancer biology, a frequent MCAT theme. Mutations in checkpoint proteins can lead to uncontrolled cell division. Another checkpoint occurs during mitosis, the spindle assembly checkpoint, which ensures that all chromosomes are properly attached to spindle fibers before separation. On exam questions, you might be asked what happens if a checkpoint fails. For example, failure at G1, S, can allow damaged DNA to be replicated, increasing mutation rates. This concept also connects to pharmacology, as many chemotherapy drugs target rapidly dividing cells by disrupting checkpoints or DNA replication. Understanding checkpoints helps you interpret experimental manipulations where certain proteins are inhibited and predict whether cells will arrest or continue dividing. Mitosis proceeds through prophase, metaphase, anaphase, and telophase, often remembered by the mnemonic PMAT, and each stage has distinct structural changes that ensure accurate chromosome segregation. In prophase, chromatin condenses into visible chromosomes, each consisting of 2 sister chromatids, and the mitotic spindle begins to form as centrosomes move to opposite poles. The nuclear envelope starts to break down, allowing spindle fibers to access chromosomes. In metaphase, chromosomes align along the metaphase plate, an imaginary plane equidistant from the 2 poles. This alignment is critical for equal division. A key MCAT detail is that each chromatid's kinetochore is attached to spindle fibers from opposite poles. In anaphase, the centromeres split and sister chromatids are pulled apart toward opposite poles, becoming individual chromosomes. This is where the actual separation of genetic material occurs. Telophase follows, where chromosomes decondense back into chromatin and new nuclear envelopes form around each set of chromosomes, essentially reversing prophase. The MCAT often tests your ability to identify stages from descriptions or images, such as recognizing metaphase by aligned chromosomes or anaphase by separating chromatids. Another common question involves errors like non-disjunction, where chromosomes fail to separate properly during anaphase, leading to aneuploidy, which connects to genetics topics like Down syndrome. Understanding PMAT also helps when comparing mitosis to meiosis, where similar stages occur, but with key differences in chromosome behavior. Mastering these stages allows you to visualize cell division and interpret experimental microscopy data. Cytokinesis is the process that divides the cytoplasm to produce 2 separate daughter cells, and it occurs differently in animal and plant cells due to the presence of a rigid cell wall in plants. In animal cells, cytokinesis involves the formation of a cleavage furrow, which is created by a contractile ring of actin and myosin filaments that pinch the cell membrane inward until the cell splits in 2. This mechanism resembles muscle contraction and is a nice connection to the cytoskeleton topic, particularly microfilaments. In plant cells, however, the rigid cell wall prevents pinching, so cytokinesis occurs through the formation of a cell plate. Vesicles from the Golgi apparatus fuse at the center of the cell, forming a growing plate that eventually develops into a new cell wall separating the 2 daughter cells. A helpful way to remember this is animals pinch, plants build. On the MCAT, you might be asked to distinguish these mechanisms or predict what would happen if actin polymerization were inhibited, which would impair cleavage furrow formation in animal cells. Cytokinesis typically overlaps with telophase, but is considered a distinct process because nuclear division and cytoplasmic division are controlled separately. Another common angle is recognizing that failure of cytokinesis can result in multinucleated cells, which has implications in certain diseases and developmental processes. This topic also connects to membrane trafficking and the role of the Golgi apparatus in vesicle formation. Understanding cytokinesis ensures you can fully account for how one cell becomes 2, completing the cell cycle. Regulation of the cell cycle by cyclins, cyclin-dependent kinases, and tumor suppressor proteins ensures orderly progression through the cycle, and disruptions in this system are a hallmark of cancer, making this a high-yield MCAT topic. Cyclins are proteins whose concentrations fluctuate throughout the cell cycle, while CDKs are enzymes that, when bound to cyclins, phosphorylate target proteins to push the cell into the next phase. Different cyclin CDK complexes act at different checkpoints, such as cyclin D, CDK4, 6' G1, and cyclin B, CDK1, for entry into mitosis. A useful mnemonic is cyclins cycle, CDKs drive. Tumor suppressor proteins like p53 and Rb act as brakes on the cell cycle. For example, p53 can induce cell cycle arrest or apoptosis in response to DNA damage, while Rb regulates the G1/S transition by controlling transcription factors. On the other hand, proto-oncogenes promote cell division, and when mutated into oncogenes, they can cause uncontrolled proliferation. The MCAT often tests scenarios where these regulators are mutated, asking you to predict outcomes such as increased division rates or failure to repair DNA. This concept ties into genetics, molecular biology, and even biochemistry through phosphorylation signaling pathways. Additionally, many cancer therapies target rapidly dividing cells by interfering with cyclin CDK activity or DNA replication. Understanding how these regulatory systems work not only helps you answer passage-based questions, but also provides a framework for thinking about disease mechanisms and treatment strategies.